Hi guys, JM.mov here, and today we're going to be looking at neurotransmitters. So this is Neuroscience 4A. Alright, now there'll be a 4B coming on later on where we'll be looking at receptors for these neurotransmitters. Alright, but today we are going to be only looking at neurotransmitters themselves. So let's see what we have covered so far. In Neuroscience 1, we looked at the structure of the neuron. Neuroscience 2, we looked at the production of an action potential. And in Neuroscience 3, we looked at the cholinergic synapse. And all these podcasts are available on YouTube. Just do a search for Biochem JM YouTube channel. Alright? So if you are watching this podcast, it should mean that you are on YouTube. So look for these other previous 3 podcasts, Neuroscience 1, 2, and 3. So today we're going to be looking at neurotransmitters. Unfortunately, we cannot look at all the neurotransmitters out there. We're going to be focusing on 3 of them. So we're going to be looking at glutamate, GABA, and finally we're going to be looking at nitric oxide. So these are the 3 neurotransmitters that we'll be looking at today. So what exactly are we going to be doing with these 3 neurotransmitters? Well, to start off, we're going to be defining what a neurotransmitter is. We're going to be looking at the criteria for accepting a substance as a neurotransmitter, the different major classes of neurotransmitters. We're going to be looking at those 3 individual neurotransmitters, and we're going to be looking at how they are synthesized, how they are released, and how they are removed. Alright, the receptors on which they act and their role in physiological functions. Well, actually, we won't be looking at receptors today so much. We're going to leave that as a separate podcast. And finally, we'll discuss the clinical conditions related to transmitter dysfunction. So first, the definition of a neurotransmitter. And a neurotransmitter is defined as a chemical substance that is synthesized in a neuron, released at a synapse following depolarization of the nerve terminal, usually dependent on influx of calcium ions, which binds to receptors on the postsynaptic cell and or presynaptic terminal to elicit a specific response. Now, if you have gone through Neuroscience 1, 2, and 3, you should know and be familiar with the term depolarization, as well as you would appreciate what happens when there's an influx of calcium ions. If not, you need to go back to Neuroscience 2, where we looked at the production of an action potential. So in that podcast, you would have be familiar with the term depolarization. And then in Neuroscience 3, when we look at the cholinergic synapse, you will see the importance of the influx of calcium ions. And just to note, we have already looked at one neurotransmitter, and that is acetylcholine. So when we looked at neurotransmitter 3, the cholinergic synapse, we looked at acetylcholine. And acetylcholine is the poster boy for neurotransmitters. We have studied that in great detail, and we know exactly how acetylcholine works. Alright. So first of all, let's look at the criteria used for identifying neurotransmitters. The substance must be synthesized in the neuron, and the enzymes needed for its synthesis must be present in the neuron. It must be released in sufficient quantity to elicit a response from the postsynaptic neuron or cell located in the effector organ. Mechanisms for removal or inactivation of the neurotransmitter from the synaptic cleft must exist. And finally, it should mimic the action of the endogenously released neurotransmitter when administered exogenously at or near a synapse. So here now are some of the major classes of neurotransmitters. So the first class there will be like, for example, acetylcholine, and this was, sorry, that should have been a box there. Yeah. So acetylcholine was discussed in Neuroscience 3 when we looked at the cholinergic synapse. We could call those, some textbooks will say those are small molecule neurotransmitters. Alright, so neurotransmitters such as acetylcholine are described as small molecule neurotransmitters. Another group would be the amino acid. Yes, in case you didn't know before, some amino acids, they actually act as neurotransmitters. And the two main amino acid neurotransmitters would be the ones that we'll be discussing today, which is glutamate, and the next one will be GABA. So those are the two we're going to be doing today. Alright, and you will see that GABA tends to be inhibitory. Alright, so gamma is what we call an inhibitory neurotransmitter, while glutamate tends to be described as an excitatory neurotransmitter. And believe it or not, these two amino acids are the main neurotransmitters for all the neurotransmission occurring in the brain. They are the workhouses, so to speak. Then we go to the monoamines or the biogenic amines, and you have dopamine, norepinephrine, and epinephrine. Alright, so those are three examples of monoamines, and we're going to be talking more of them in later podcasts. Then we have the peptide neurotransmitters, such as the endorphins and neuropeptide Y. And then we have those that really don't fall under the previous groups. We say that they are under other, and you have adenosine, which is an interesting one. Alright, and nitric oxide, that simple gas has a lot of potential. It does a lot of things in the body. Right, so let's start with glutamate. Now, as I said before, glutamate is an excitatory neurotransmitter. It occurs in high concentrations in the brain, and although it plays a role in other functions such as the synthesis of proteins and peptides, glutamate fulfills the criteria for a substance to be accepted as a neurotransmitter. And basically what I'm saying is that this would not be the first time you are hearing about glutamate. You would have come across glutamate previously, and glutamate would have been playing a different role. But it also fulfills the criteria for a neurotransmitter, and we consider glutamate a neurotransmitter. Alright, so now glutamate is a typical, it falls under all the criteria of a typical neurotransmitter. In other words, if you have already studied the cholinergic synapse, which was in Neuroscience 3, and if you know how the mechanism behind that, it is a similar mechanism to what's happening in the glutamate transmission as well. So it's the same thing as the cholinergic transmission is the same thing as what's happening here. Same kind of general mechanism. Alright, so let's look at what's happening here. So again, you would have your pre-synaptic cell, you have your post-synaptic cell, and you have a glial cell. And I think I asked you before to do some research and find out what a glial cell is. So you should know what is the function of a glial cell now. Now let's look at, they have numbered the steps. Let's talk about it from the way they have done it. So the glutamate, which is in the synaptic cleft here, some of it, the glutamate molecules, could be taken up by these glutamate transporters. So glutamate can be taken up by the presynaptic terminal, or it can be taken up by the glial cell, both using glutamate transporters. Now when glutamate enters the glial cell, the glutamate is converted to glutamine via an enzyme glutamine synthetase. So note these enzymes. This is happening in the glial cell. So let's label that GL, glial cell. Alright. So glutamate is being converted to glutamine using glutamine synthetase. Alright. And there's a glutamine transport on the glial cell where the glutamine could leave the glial cell now, and it could go to the presynaptic cell. So glutamine will enter the presynaptic cell via these glutamine transporters. Now once glutamine enters the presynaptic cell, it is converted back to glutamate. Now something to note is that the enzyme that converts glutamine to glutamate in a presynaptic cell, it's a mitochondrial enzyme, but that mitochondrial enzyme is named glutaminase. So be careful about that. That could be a trick question in the exam. So be careful. Glutaminase will convert glutamine to glutamate, right, in the presynaptic cell. In other words, we don't use back the same glutamine synthetase and do a reverse reaction. Alright, so be careful about these two guys here. Know the location, right? So glutamine synthetase is in a glial cell and converts glutamate to glutamine. And if you want to convert glutamine back to glutamate, you use a mitochondrial enzyme called glutaminase, which is found in a presynaptic cell. Now once the glutamate is produced, it's packaged and stored in vesicles. Alright, so vesicles will contain these glutamate neurotransmitters stored. When there's an influx of, when there's a wave of depolarization, they will have an influx of calcium ions. And when there's that influx of calcium ions, these vesicles, they fuse to the presynaptic membrane, and the glutamate, which is a neurotransmitter, is released via exocytosis. And then this glutamate will cross the synaptic cleft and bind to its receptor. Alright, so glutamate will bind to the receptor and the ion channel will open and let ions come in. Alright, and if you looked at cholinergic synapse in Neuroscience 3, you'll realize that there's a lot of similarities here. The mechanism is the same, just that you're going to be changing the enzymes and so on. But the mechanism is basically the same. So some clinical considerations now. Glutamate has also been implicated in some chronic neuropathological conditions such as amyotrophic lateral sclerosis, otherwise known as ALS, which is characterized by degeneration of the motor neurons in the anterior horn of the spinal cord, brainstem, and cerebral cortex. And what that causes is that patients suffering from this disease exhibit widespread muscle twitching, weakness, and atrophy of the muscles. Alright, so that's a sideline there. Now we go to the second neurotransmitter, which is GABA. And GABA stands for gamma aminobutyric acid. And GABA is found in high concentrations in the brain and spinal cord, but is absent in peripheral nerves or peripheral tissues. Unlike glutamate, GABA is not an essential metabolite, and it is not incorporated into a protein. So glutamine is converted into glutamate by an enzyme glutaminase. GABA is formed by the decarboxylation of glutamate. This reaction is catalyzed by a cytosolic enzyme, which is called glutamate decarboxylase, or GAD, which is present almost exclusively in GABAergic neurons. GAD is not present in neurons using glutamate as transmitter or in glial cells. Alright? So the glutamate decarboxylase is not present in neurons using glutamate as transmitter, as a neurotransmitter, or in glial cells. Glutamate decarboxylase requires pyridoxal phosphate, or in other words PLP, as a coenzyme. Alright, so just remember that is a very popular question in the exam. What is the coenzyme for glutamate decarboxylase? The answer is PLP, pyridoxal phosphate. And you get PLP from vitamin B6. Right, so if you look at the diagram there, you see that glutamate, which is this guy here, glutamate is the precursor molecule for GABA. We're going to use glutamate to make GABA. And the enzyme that catalyzes this reaction is called glutamate decarboxylase. And it uses PLP as its coenzyme. Now, if you watch the difference, between these two molecules, between glutamate and GABA, you realize that we lose this COO minus group here. Alright, this COO minus group is lost as carbon dioxide. And that is why we call it a decarboxylation reaction, because you are losing carbon dioxide. And we call it gamma aminobutyrate. Alright, because it's but because it has 1, 2, 3, 4 carbons. The amino part will be this group here, the NH3+, and it's gamma because the carbon that's attaching the amino group is the gamma carbon. Alright, so the gamma carbon here is what the NH3+ is attached to. So therefore we call it a gamma amino butyrate because there's 1, 2, 3, 4 carbons. Okay, so that's what GABA gets its name. And this is a decarboxylation reaction that is catalyzed by glutamate decarboxylase. And remember, what's the coenzyme for glutamate decarboxylase? PLP. Alright, so let's look at the general mechanism here. And once again, it's very similar to the cholinergic synapse, that mechanism. So if you watch, let's find one, step 1, that is, and that's here. So we're using the mitochondrial enzyme glutaminase to convert glutamine to glutamate. This glutamate now is then converted to GABA using the cytosolic enzyme. But they have glutamic acid, but you know glutamic acid is another word for saying glutamate. It's the same thing. So glutamate decarboxylase, in other words, the GAD enzyme here. Alright, so let me just write that here. So GAD. Alright. Once GABA is synthesized, the GABA is stored in vesicles. And there's a transporter protein here that could bring in the GABA and let it be stored in these vesicles. When there's a wave of depolarization on this presynaptic neuron, alright, there will be an influx, once again, of calcium ions. And that will cause the release of GABA via what process? It's the same process as in glutamate as well as in the cholinergic synapse. And that will be correct. You'll be saying exocytosis. Alright. So the GABA is released into the synaptic cleft. The GABA will then bind to its receptor on the postsynaptic cell, and an ion channel is going to open. The GABA will then be either reabsorbed via the absorbed by the presynaptic neuron, or it can be absorbed by the glial cells via GABA transporters, right? So you have GABA transporters where the GABA can be reabsorbed by the presynaptic neuron as well as the glial cell. Now when GABA enters the glial cell, it is converted to glutamate, and the enzyme that is being used there is, well, they have here GABA T, alright, which is just saying GABA transaminase. So the enzyme that converts GABA to glutamate in the glial cell is called GABA transaminase. So that converts the GABA to glutamate. The glutamate is then converted to glutamine using the enzyme glutamine synthetase. Once you have glutamine, it passes through the glutamine transporters, alright, and then it will enter the presynaptic neuron via its glutamine transporters. And once it reaches here, it then goes through the process again, where the glutamine will be converted to glutamate and then to GABA and so on. Now, the GABA neurotransmitter is inhibitory. Now, before I even tell you why, I want you all to try to figure it out. Propose some mechanisms by which a neurotransmitter could have an inhibitory effect. Because so far, when we looked at the cholinergic synapse in Neuroscience 3, and now when we looked at the glutamate transmission just before this one, we saw that both of them were excitatory. And so therefore they would have certain things happening. But GABA is inhibitory. So kind of try to figure out and devise a possible mechanism in which GABA can be inhibitory. So write down something and then we will continue. So I'm waiting on you patiently to think of something. Alright? So yeah, the receptors for these substances, so in other words, the GABA receptor on the post-synaptic cell, right, are ligand-gated channels that are selectively permeable to Cl- chloride ions. So in other words, when GABA binds to the receptor on the postsynaptic cell, ligand-gated channels, but ligand chloride-gated channels, they open, and chloride ions, Cl- ions comes in. So let me ask you a question before we even move on. If chloride ions are coming in, what's going to happen to the membrane potential? Alright? And the hint here is to look at the charge of the chloride ions. So that should tell you what's going to happen to that membrane potential. So hence, the opening of these chloride ion channels, it tends to hyperpolarize the membrane. Alright? And you should know this term hyperpolarize from your action potential podcast, which was Neuroscience 2. In Neuroscience 2, we spoke about this term hyperpolarize. And hyperpolarize means that the membrane potential becomes more negative. And you can see why, right? It is because negative ions, chloride ions, is coming in. Now, normally, it would have been a depolarization. But a depolarization would have been caused because there were Na+ ions coming in. But that is not what this neurotransmitter is doing. This neurotransmitter is not like acetylcholine or glutamate, where when the ion channel is open, Na+ comes in. No, GABA is inhibitory. It opens chloride ion channels. So Cl-, negative ions is coming in. And when negative ions come in, it's going to make the inside the membrane, which was originally negative at rest. Alright, so the resting potential was already negative around -70 millivolts. And if you have negative ions coming in now, it's going to even make it more negative. So we say that that's hyperpolarized. So a neuron inhibited in this manner must therefore be more intensely depolarized than otherwise to trigger an action potential. In other words, because of this hyperpolarized state, it's even more negative. So it's going to take, it's going to take a really strong stimulus, alright, to make this membrane become depolarized enough for an action potential to be generated. So that's why we're saying GABA has an inhibitory effect. It makes it very difficult, because of its hyperpolarized state, for an action potential to be developed. So there's a little summary there. Anion channels, and remember anion will be negative ions such as chloride ion, Cl-. So anion channels are inhibitory, whereas cation, and cation is positive ions such as Na+, cation channels are excitatory. And this on a side note, ethanol, the oldest and most widely used psychoactive drug, is thought to act by inducing GABA receptors in the brain to open their Cl- or their chloride ion channels. The GABA A receptor channel is pharmacologically important because it is the target of Valium, which is used to diminish anxiety. So a lot of these anti-anxiety drugs, they target the GABA A receptor. All right. All right. So we looked at glutamate. We looked at GABA. Now I just put this slide to tell you how fascinating biochemistry is. All right. So at this point now we have looked at glutamate in different things. So this course is a level 3 biochemistry course. So remember that you have done a lot of biochemistry along the way, and you should appreciate now that glutamate can be a neurotransmitter. It is the precursor molecule to make GABA. Alright, you use glutamate to make N-acetylglutamate, and you should know that that is a very powerful activator of CPS1, which is the key enzyme in urea cycle. Glutamate is key in transamination reactions as well as oxidative deamination. And when we do sensory systems, you'll see that glutamate plays a very important role in vision as well as taste. So you see how versatile this glutamate molecule is. And you see this a lot in biochemistry. You see that certain molecules, they have so many different roles, and you know, it's only when you cover a lot of biochemistry and then you start looking at it from the outside, you're going to see all these links, right? So as a teacher, you know, I can't wait for that day when your biochemistry kung fu is sharp enough that you can make these kind of links. So it's not only glutamate, you're going to see other things integrated into a lot of other topics. Like for instance, before we had this lecture today, I'm sure you wasn't even thinking that glutamate, a simple small little amino acid, could act as a neurotransmitter, but yet it does. Alright? And then you would never thought that glutamate plays a role in taste and vision, but it does. Alright? So it's a really fascinating world out there. Biochemistry, it's fascinating, it's interesting, it's sexy, you know, and just keep on reading. There's so much out there, right? And there's so much that we don't know as yet. So who knows, one of you guys who's listening right now might be the one who to write the next best chapter out there, and please just send me a copy so I can take a good read. So the last and final neurotransmitter that we'll be looking at today will be the gaseous neurotransmitter. So it's a gas. And the gas I'm going to be looking at is nitric oxide. Now this is a new class of neurotransmitters, and it doesn't fall under the classical concept of a neurotransmitter, and we will see that just now. Now nitric oxide and carbon monoxide are two important members of this class. Nitric oxide is discussed as a representative of this group because relatively more information is available for this neurotransmitter. In isolated vascular smooth muscle preparations, acetylcholine and other vasodilators release a short-acting substance from the endothelial cells that relaxes blood vessels. And for a long time, we thought that this relaxing factor was endothelium-derived relaxing factor. But after, you know, some further studies and so on, we realized that this EDRF that we were thinking is some separate entity is actually and simply nitric oxide. Right? So nitric oxide was this molecule that we were naming EDRF. So it is now well recognized that nitric oxide plays an important role in mediating vasodilation. Nitric oxide is also produced in many other cells, including neurons, where it has been implicated as a neurotransmitter. So the major effect of nitric oxide in the body is vasodilation. Alright, and it's fascinating, you know, because you know, in further reading you're going to see a lot of cool stuff. Like for instance, you'll see that in diabetics, they tend to have lower than normal levels of nitric oxide, and we believe that is why they have so many circulatory problems. Alright, they have poor circulation in the eye as well as in the heart and the kidneys, and that's why they get problems. They get, you know, problems with the eyes and kidneys and heart because of this poor circulation. So definitely nitric oxide has a role there. You would also see that nitric oxide plays a role in bringing the dead alive, so to speak. What we thought was dead for many years soon becomes alive. And I'm making reference to Viagra, right? And everybody knows what Viagra does, right? And the key component there is, again, nitric oxide and its role in vasodilation. So you can do some reading up about that. So the formation of nitric oxide, because this is our biochemistry course, we are interested in these kind of things. Alright? So you see that the molecule that we use for nitric oxide, the precursor molecule here will be arginine. Alright? And arginine will be converted to citrulline and nitric oxide. And the enzyme that catalyzes this reaction is called nitric oxide synthase. So easy to remember. So if you want to increase the amount of nitric oxide being produced in your body, you could increase the intake of arginine, because that's going to be converted to citrulline and nitric oxide. Alright, now some other things here to note. You will need 2 molecules of oxygen, this one and this one, to make citrulline, as well as you will need 2 molecules of NADH. Alright, and that will produce 2 molecules of water as the products. Okay, and let's note that the enzyme is nitric oxide synthase. So this is one of the times where biochemistry has been very kind to us with the naming of the enzyme. So again, the enzyme that synthesizes nitric oxide is called nitric oxide synthase. You can't get easier than that. So let's look at what's happening here. Now, something I want to say up front is that in nitric oxide transmission, neurotransmission, scientists will say that, it's being produced by nerve cells, and it is having a physiological effect. So by that definition, it's a neurotransmitter. But if you go back to the previous slides where we look at the criteria for a neurotransmitter, it does not fulfill all of them. And a common exam question is for you to state how nitric oxide is not really behaving as a neurotransmitter. So hopefully after we discuss what's happening here, you should be jotting down some points about how it goes against the classical neurotransmission, like for instance in a cholinergic synapse. Alright? So what I want you all to do is to keep in mind the cholinergic synapse where you had acetylcholine neurotransmission, and compare that to this nitric oxide neurotransmission, and tell me how they are different, because they are different. The mechanism is different. Alright, so let's see where they start. They started with glutamate. So this is step 1. Now glutamate is going to bind to its NMDA receptor. So it binds to its NMDA receptor. So like this guy here. Now when glutamate binds to its NMDA receptor, that receptor undergoes a conformational change that will allow calcium ions to come in. Alright? So calcium ions, they come in. Now with that influx of calcium ions into the postsynaptic neuron, alright, that calcium ions will bind to a calcium binding protein that we call calmodulin. Alright, so this influx of Ca2 plus ions, calcium ions, these calcium ions bind to a protein called calmodulin. And this calcium calmodulin complex will then activate nitric oxide synthase. And the nitric oxide synthase is going to convert the arginine to citrulline plus nitric oxide. That's that reaction we just looked at. Alright, so again, nitric oxide synthase is converting arginine to citrulline plus nitric oxide. Alright, now you're seeing this N here, that is there are different isozymes or isoforms of nitric oxide synthase. So the one that we're going to be looking at is the N form. You You can read up more about that later on. But this nitric oxide, it interacts with the heme moiety of a soluble guanylate cyclase enzyme. So again, the nitric oxide will interact with the heme moiety of soluble guanylate cyclase. And when that is activated, all right, via allosteric transformation, all right, there's an activation of this enzyme, it produces cGMP. And when there is increased levels of cGMP in the postsynaptic neuron, it results in a physiological response. So there's a physiological response when the cGMP levels increase. All right. So when the cyclic guanosine monophosphate levels, when they increase, there's a physiological response. Now here's the thing. This nitric oxide is a very small molecule. All right, it's nonpolar. It's small. And what will happen is that it could go through this bilayer here very easily in the postsynaptic neuron, and then it could go into adjacent cells, whether it be a adjacent neuron or a glial cell. It could go into both of them. All right, so it diffuses over to adjacent cells, which could be either neurons or glial cells. Now, when they diffuse there, diffusion of the nitric oxide, all right, it stimulates again, just like how it did in this postsynaptic cell. It will stimulate the soluble guanylate cyclase, and it will increase cGMP levels once again, and that will lead to a physiological response. All right. Now that will continue until well, what happens is that the nitric oxide, it has a short half-life, and it decays spontaneously and is converted to nitrites and nitrates, oxygen and water. All right. So it doesn't stay there forever. It breaks down spontaneously. So there's no extra enzymes or anything that is needed to break it down. It breaks down. We say it's a passive process. Nothing is needed to do it. Oh, one last thing. The nitric oxide, when it diffuses out of this postsynaptic cell, it could have gone to adjacent neurons. It could have gone to the glial cell, and it could also diffuse into the presynaptic cell. All right. So the nitric oxide could also diffuse, all right, into the presynaptic neuron. So that is just something else that I was supposed to point out. So again, what you could do now is to go over what I just said and see how this is different from a cholinergic synaptic transmission. Because there's a few things that are being done here that is different. So you see here I said that nitric oxide is described as a retrograde messenger. And I kind of hinted to it in the last slide, but I want you to do all the homework. Tell me and explain what is meant by a retrograde messenger. Why am I saying that nitric oxide is a retrograde messenger? Right, so remember I was saying that nitric oxide, it differs from the classical neurotransmitters such as acetylcholine, for instance. So let's read. Nitric oxide does not satisfy some of the criteria formulated for classical transmitters, as it differs from conventional neurotransmitters in the following respects. First of all, it is not stored in vesicles and is generated only when it is needed. So if you look at acetylcholine and if you look at glutamate and GABA, they were all stored in vesicles. We don't have vesicles containing nitric oxide. That is probably a good thing because nitric oxide is very reactive and that could have caused a lot of problems for the cell. It is not released by calcium-dependent exocytosis from a presynaptic terminal. So that doesn't happen. All right, it isn't like with, for instance, like with acetylcholine where there's an influx of Ca2+. in the presynaptic terminal, and that will cause a release of the neurotransmitter. No, that is not what is happening here. Nitric oxide is an uncharged molecule. It diffuses freely across cell membranes and modifies the activity of other cells. So remember I was saying this before, inactivation of nitric oxide is passive. And what that means, there is no active process that terminates its action. So like for instance, like with acetylcholine. And I'm always going back to acetylcholine because acetylcholine is the poster boy when it comes to the classical neurotransmitter and how it works. Right? This is one of the first neurotransmitters that we really studied, and therefore it's always one that we refer everything to. All right, and you see here that what we saw in Neuroscience 3 was that acetylcholine is broken down by an enzyme called ACHE, all right, which is acetylcholine esterase. Here we don't need such an enzyme. What happens to the nitric oxide is that it decays spontaneously. It decays and is converted to nitrites and nitrates, oxygen and water. And next thing, it does not interact with receptors. So if you realize, that was something strange, eh? The nitric oxide, because it's small, like it just diffuse into any neighboring cell. It doesn't need a receptor, all right? Its sphere of action depends on the extent to which it diffuses, which may include several target cells. Therefore, the action of nitric oxide is not confined to the conventional presynaptic to postsynaptic direction. Because if you look at acetylcholine, you look at glutamate, you look at GABA, we saw that it starts off in the presynaptic cell and then it goes over to the postsynaptic cell. That's the classical direction. What we're saying is that for nitric oxide, that's not happening. Right? It's a totally different thing. It's not that conventional direction. And lastly, nitric oxide acts as a retrograde messenger and regulates the function of axon terminals presynaptic to the neuron in which it is synthesized. And I remember I told you, for your homework, I want you to just give me some more information about what a retrograde messenger is. Right, and you might be saying, man, this man only busting homework material. But it's just for you to get a better understanding of the lecture, as well as I shouldn't be giving you all everything. I want you to go and explore some of these things. So by looking at the mechanism of action and stating the uses of these following, you get a better feel of, you know, what neurotransmitters do and how blocking or stimulating their receptors, what effect they could have in medicine and so on. Alright, so do some little homework. It's very interesting that you read up about these guys. So give it a little read. You know, you never know, you might sit in an exam. Who knows? Now, we have looked at GABA, we looked at glutamate, and we finally look at nitric oxide. Now, there are over, I believe, between 30 to 50 neurotransmitters that we have discovered so far. And obviously, we can't do each one. And what scientists believe is that we suspect there's a lot more of neurotransmitters that we have not discovered as yet. Alright? Now, these three neurotransmitters here are very sexy. They're very popular. Alright? And we're going to be looking at them later on. So I just thought I should just mention them here. So we have not covered all the neurotransmitters. We can't. We just covered some. But I will tell you all, rest assured, we're going to be covering these neurotransmitters later on. So like for instance, dopamine. Alright, dopamine. We're going to be covering dopamine actually in the next set of lectures coming up, where we look at Parkinson's disease. Alright, as well as the role of dopamine in schizophrenia. So I'll just put schiz here for now. Serotonin, we will look at later on. Alright, how levels of serotonin could have an impact on depression. Alright, the whole word as a starter. And we will look at substance P under sensory systems. And substance P plays a key role in pain perception. Alright, so we're going to be coming across these guys later on. So again, dopamine, it plays a big role in Parkinson's disease as well as schizophrenia. Serotonin as well as norepinephrine, will be play an important role in depression, and substance P, which is a neuropeptide, it has a big role to play in pain perception. So when we're doing sensory system and we're looking at pain, we'll talk a little bit about substance P. So again, let's repeat, in the next class, we're going to be studying Parkinson's disease. So I'm going to tell you a lot about Parkinson's disease. As usual, guys, I really hope that you all learned something here. If you did, and you like what you got, please show your love and hit the like button, as well as subscribe to the channel, because I'll be producing more and more videos and I want you to get them as soon as they are uploaded. Alright? And as usual, I would love to get feedback from you all. This is my my Gmail address. Please send me any of your comments, as well as if you all have any cool diagrams and pictures that you all found that was helpful for you all in learning this topic. Please share it with us so I can share it with my students. And as usual, thank you a million for listening to this podcast. I really hope it helps. And just keep studying. Biochemistry could be a little intimidating, I will admit, but it is so interesting. And you know, just give it some time and you'll see. You're gonna have a lot of fun with this topic. Alright guys, so best of luck in your studies and we'll talk soon.